Escherichia coli, commonly known as E. coli asterisk, is a type of bacteria that resides in the intestines of humans and animals. Discovered by the German pediatrician and bacteriologist Theodor Escherichia in 1885, E. coli asterisk has since become one of the most well-researched organisms in microbiology. Its name honors Escherichia and the Latin term, coli, referring to the colon, where it is primarily found. This rod-shaped bacterium is classified as gram-negative due to its cell wall structure, and it is often non-pathogenic. However, some strains of E. coli can cause severe illness in humans. E. coli plays a crucial role in the gut microbiome, contributing to the digestion process and the synthesis of essential vitamins like vitamin K. It is a facultative anaerobe, meaning it can thrive in both oxygen-rich and oxygen-depleted environments, allowing it to adapt easily to varying conditions. E. coli reproduces rapidly through binary fission, where a single bacterium divides into two, facilitating the quick colonization of new environments. Despite its beneficial aspects, E. coli is perhaps best known for its pathogenic strains. These strains have acquired virulence factors that enable them to cause a range of diseases, from gastrointestinal infections to life-threatening conditions like hemolytic uremic syndrome, HUS. Understanding the dual nature of E. coli asterisk, both as a harmless gut bacterium and a potent pathogen, is essential for comprehending its impact on health and disease. Not all strains of E. coli are harmful. In fact, most strains coexist peacefully within the human intestines. However, certain strains have evolved to become virulent pathogens, capable of causing severe illness. These pathogenic strains are broadly classified into 6 major groups: enterotoxigenic E. coli, ETEC, enteropathogenic E. coli, EPEC, enteroinvasive E. coli, EIEC, enterohemorrhagic E. coli, EHEC, enteroaggregative E. coli, EAEC, and diffusely adherent E. coli, DAEC. Each group is associated with specific types of infections, symptoms, and mechanisms of action. One of the most dangerous strains is E. coli O157, H7, which falls under the enterohemorrhagic category. This strain produces a potent sugar toxin that can damage the lining of the intestines, leading to bloody diarrhea, abdominal cramps, and vomiting. In severe cases, it can cause hemolytic uremic syndrome, HUS, a life-threatening condition that results in kidney failure, especially in young children and the elderly. Enterotoxigenic E. coli, ETEC, is another significant strain, commonly known as the cause of traveler's diarrhea. It releases 2 types of toxins that affect the intestines, leading to watery diarrhea, dehydration, and electrolyte imbalance. Though ETEC is rarely life-threatening, it can cause severe discomfort and complications, especially in regions with poor access to clean water. Each strain of pathogenic E. coli asterisk has a unique way of entering the body, whether through contaminated food, water, or person-to-person contact. The ability of these strains to acquire and transfer virulence genes through plasmids, transposons, or bacteriophages makes E. coli a versatile and evolving pathogen that poses ongoing public health challenges. E. coli is commonly found in the environment, in soil, water, and the intestines of warm-blooded animals. While most strains are harmless, the transmission of pathogenic E. coli occurs through several routes, making it a significant public health issue. Contaminated food, particularly undercooked meat, especially ground beef, raw vegetables, and unpasteurized dairy products, are among the most common sources of E. coli infections. Waterborne transmission is also a concern, especially in regions with poor sanitation. Food safety plays a critical role in controlling E. coli outbreaks. The bacteria can survive in a wide range of conditions, from acidic environments in the stomach to the cold temperatures in refrigerators. Improper food handling, such as inadequate cooking or cross-contamination of surfaces, allows E. coli to spread and infect new hosts. The large-scale distribution of contaminated food products can lead to widespread outbreaks, affecting thousands of people across different regions. The epidemiology of E. coli also reveals its presence as a zoonotic bacterium, meaning it can be transmitted between animals and humans. Farm animals, particularly cattle, are known reservoirs for pathogenic strains like O157, H7. When fecal matter from these animals contaminates water sources or food, it increases the risk of human infection. International travel and global food distribution further complicate the containment of E. coli outbreaks, allowing the bacteria to cross borders and infect populations in distant locations. Public health agencies continuously monitor and investigate E. coli outbreaks, utilizing molecular tools such as whole genome sequencing to track bacterial strains and identify sources of contamination. Despite these efforts, the emergence of antibiotic-resistant E. coli strains poses a growing threat to treatment and prevention strategies. Symptoms of E. coli infection can vary depending on the strain and the health of the individual. Gastrointestinal symptoms are most common and typically begin within a few days of exposure. These symptoms include abdominal cramps, diarrhea, which may be bloody, nausea, vomiting, and sometimes fever. Severe cases, particularly with strains like O157, H7, can lead to complications such as hemolytic uremic syndrome, HUS, which requires immediate medical attention. Diagnosis of E. coli infections usually involves laboratory tests that identify the bacteria in stool samples. Rapid diagnostic techniques, including polymerase chain reaction (PCR) tests, can detect specific pathogenic strains by targeting their virulence genes. In some cases, culture methods are used to isolate the bacteria and perform antimicrobial susceptibility testing, which helps determine appropriate treatments. Treatment for E. coli infections depends on the severity of the symptoms. For mild cases, supportive care such as hydration and rest is typically sufficient. Antibiotics are not recommended for certain strains, particularly O157, H7, as they can increase the risk of HUS by causing the bacteria to release more toxins. In severe cases, hospitalization may be necessary to manage complications like kidney failure or dehydration. The emergence of antibiotic-resistant E. coli strains complicates treatment, making some infections harder to control. Research into new therapeutic approaches, including bacteriophages, viruses that target bacteria, and vaccines, offers hope for more effective treatments in the future. Preventing E. coli infections requires a multifaceted approach that includes personal hygiene, food safety practices, and public health measures. One of the most effective prevention strategies is proper hand washing, particularly after using the bathroom, handling raw meat, or coming into contact with animals. This simple practice can significantly reduce the spread of E. coli and other pathogens. In the food industry, stringent safety standards are essential to prevent contamination. Cooking meat to the recommended internal temperature, washing fruits and vegetables, and avoiding cross-contamination between raw and cooked foods can help minimize the risk of E. coli infections. Pasteurization of dairy products and proper water treatment are also critical in preventing outbreaks. On a broader scale, public health agencies play a vital role in controlling E. coli* by monitoring outbreaks, issuing food recalls, and educating the public about safe practices. These agencies collaborate with food producers, distributors, and healthcare providers to ensure rapid responses to potential threats. However, the rise of antibiotic-resistant strains highlights the need for ongoing surveillance and research to develop new treatment options and containment strategies. Ultimately, while E. coli is a normal and often beneficial part of the human microbiome, its pathogenic strains remind us of the delicate balance between health and disease. Understanding this bacterium's biology, transmission, and potential for harm is crucial in maintaining public health and preventing future outbreaks. One of the most remarkable aspects of Escherichia coli is its widespread use in biotechnology and molecular biology. Due to its rapid growth, well-understood genetics, and ease of manipulation, E. coli* has become the workhorse of genetic engineering, cloning, and biopharmaceutical production. Scientists routinely use E. coli as a model organism to study fundamental biological processes and to produce a variety of recombinant proteins, including insulin, growth hormones, and vaccines. E. coli is particularly valuable in recombinant DNA technology. researchers can introduce foreign genes into E. coli cells, enabling the bacteria to produce proteins from other organisms. This has revolutionized medicine by making it possible to manufacture human proteins in large quantities. For instance, the production of human insulin using genetically engineered E. coli * was one of the earliest and most successful applications of this technology. Before this innovation, insulin for diabetics was extracted from the pancreases of pigs or cattle, which was less efficient and sometimes caused allergic reactions. The bacterium's role extends to the study of gene expression, gene regulation, and protein synthesis. In the laboratory, researchers use strains of E. coli that have been genetically modified to delete specific genes, insert new ones, or track how genes function under different conditions. E. coli also serves as a model for studying antibiotic resistance mechanisms, helping scientists understand how bacteria evolve in response to selective pressures like antibiotic use. This insight is critical for developing new antibiotics and combating the global issue of drug-resistant infections. Additionally, the field of synthetic biology often relies on E. coli as a platform for building novel biological systems. By altering E. coli's metabolic pathways, scientists can create strains that produce biofuels, biodegradable plastics, and other valuable chemicals. This further underscores the versatility and importance of E. coli beyond its role as a human pathogen. One of the most concerning developments in modern medicine is the rise of antibiotic-resistant bacteria, and E. coli is no exception. While antibiotics have been life-saving drugs for decades, their overuse and misuse have led to the evolution of resistant strains, some of which are extremely difficult to treat. E. coli * has become one of the bacteria most frequently associated with antibiotic resistance, particularly in hospital settings, where resistant strains like extended-spectrum beta-lactamase, ESBL, producing E. coli have emerged. Antibiotic-resistant E. coli infections pose a significant challenge for healthcare providers because they limit treatment options. In cases where traditional antibiotics such as penicillin or cephalosporins are ineffective, doctors may need to resort to last-line treatments like carbapenems or even experimental drugs. However, even these powerful antibiotics are becoming less effective due to the rise of carbapenem-resistant Enterobacteriaceae present, which includes strains of E. coli. The spread of resistant E. coli strains is fueled by several factors, including the overprescription of antibiotics, the use of antibiotics in agriculture, and poor infection control practices in healthcare settings. E. coli can acquire resistance genes through horizontal gene transfer, sharing them with other bacteria and rapidly disseminating resistance traits within microbial communities. Combating antibiotic resistance requires a global effort, involving improved diagnostic tools, responsible antibiotic stewardship, and the development of new antibiotics or alternative therapies such as phage therapy. Understanding how E. coli evolves and acquires resistance is key to devising strategies to mitigate the spread of these dangerous superbugs. In recent years, the gut microbiome has emerged as a major focus of research, revealing the complex ecosystem of microorganisms that reside within our digestive tract. Escherichia coli is one of the many species that contribute to this diverse microbial community. While pathogenic strains of E. coli can cause harm, the non-pathogenic strains serve important functions in maintaining gut health. Non-pathogenic E. coli is a commensal bacterium, meaning it coexists with the host without causing harm. It helps maintain the balance of the gut microbiota by competing with harmful bacteria for nutrients and space. E. coli also stimulates the immune system, promoting a healthy response to potential pathogens and helping the body develop tolerance to non-threatening microbes. Recent studies have shown that disruptions in the gut microbiome, often caused by antibiotic use, can lead to dysbiosis, a state of microbial imbalance associated with various health issues, including inflammatory bowel disease, IBD, irritable bowel syndrome, IBS, and even metabolic conditions like obesity and diabetes. The delicate balance of microbes in the gut, including E. coli * plays a critical role in maintaining overall health. Probiotics and prebiotics have become popular strategies for promoting a healthy gut microbiome. However, E. coli itself has been engineered as a probiotic. Strains like E. coli Nissl 1917 have been used in Europe for decades to treat gastrointestinal disorders, demonstrating the bacterium's potential as a therapeutic agent. The relationship between E. coli and the gut microbiome highlights the importance of microbial diversity and balance in health and disease. Ongoing research into how E. coli interacts with other gut microbes and the human immune system could lead to new treatments for a variety of health conditions. Beyond its role in human health, E. coli is also a significant environmental organism. It is commonly used as an indicator of water quality in environmental monitoring because its presence in water sources often signals fecal contamination. Since E. coli is found in the intestines of warm-blooded animals, its detection in water suggests that human or animal waste has entered the water system, bringing the potential for pathogens along with it. Waterborne E. coli can be transmitted through drinking contaminated water, swimming in polluted lakes or oceans, or consuming produce irrigated with contaminated water. The presence of pathogenic E. coli strains in water can lead to outbreaks of diseases such as gastroenteritis, especially in communities with inadequate sanitation infrastructure. Testing for E. coli * in water samples is a standard practice in environmental health. Public health agencies use these tests to ensure that drinking water is safe and to identify potential sources of contamination, such as agricultural runoff, sewage leaks, or improperly treated wastewater. In some regions, especially developing countries, waterborne E. coli infections remain a major cause of morbidity and mortality, particularly among children. Efforts to improve water quality and sanitation are critical for preventing E. coli outbreaks. These include the implementation of water treatment systems, regular monitoring of water sources, and the promotion of hygiene practices such as hand washing and proper waste disposal. Understanding the role of E. coli * in the environment is crucial for protecting public health and ensuring access to clean water. As one of the most studied organisms in science, Escherichia coli continues to reveal new insights into microbiology, genetics, and evolution. Its relatively simple genome and fast replication rate make it an ideal subject for experimental evolution studies, where researchers observe how E. coli adapts to different environments over multiple generations. One famous example of this is the long-term evolution experiment conducted by evolutionary biologist Richard Lenski, which began in 1988 and continues to this day. In this experiment, E. coli populations are grown under controlled conditions, with samples taken periodically to observe genetic changes over time. Lenski's work has provided invaluable insights into how bacteria evolve, adapt, and acquire new traits, an understanding that has implications for everything from antibiotic resistance to industrial applications. Looking to the future, E. coli will likely remain at the forefront of research into synthetic biology, evolution, and biotechnology. Scientists are exploring ways to engineer E. coli to perform even more complex tasks, such as breaking down environmental pollutants, producing sustainable biofuels, or acting as living sensors that detect harmful substances in the environment. However, the ongoing evolution of pathogenic strains poses a continuous challenge. The rapid rate at which E. coli can acquire new genes through horizontal gene transfer means that it has the potential to evolve new virulence factors or antibiotic resistance mechanisms. This highlights the importance of continued surveillance, research, and innovation to stay ahead of the evolving threats posed by this versatile bacterium. In conclusion, Escherichia coli remains a paradox in the world of microbiology, both a friend and foe. Its role as a vital part of our gut microbiome, a tool in biotechnology, and a persistent pathogen ensures that it will continue to be a subject of fascination and study for generations to come.